You're at the cutting edge of cardiovascular sciences. I'm your host, John Cook. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist Research Institute. And we've got a very special guest today. But before I introduce Dr. Poo, I'd just like to turn for a moment to a meeting in the future that we're going to be having here in Houston. It's the best cardiovascular bioengineering symposium of the year. It's the 8th annual cardiovascular bioengineering symposium. This meeting has grown out of two NIH-funded consortium, the Progenitor Cell Translational Consortium and the Progenitor Cell Biology Consortium. And this meeting has grown out of that, and we've had a lot of fun with it. This will be the 8th year, and we have just spectacular speakers. It's going to be May 22nd to 25 this year. And let me just scroll through the program here very quickly for you. Jay Zhang from University of Alabama is doing some great things there as chair of biomedical engineering. And Rod Pettigrew is here. He started the EnMed program, Engineering in Medicine program, the second of its kind to train engineers to be physicians, to train those physicianeers to make useful things that are going to be transformational in medicine. And then we've got some great speakers. We've got Victor Zhao coming to give the keynote. He, as you know, is the president of the National Academy of Medicine. And we have Gordana Vunjak-Novakic, who's also a star in biomedical engineering, who'll be speaking. Victor will be talking about mRNA therapeutics in cardiovascular medicine. Francisco Altamirano is chairing the first session on bioengineered translational model systems. We're going to be hearing about engineered heart tissues. We have Alessandro Grattoni, who's our chair of nanomedicine, who's going to be introducing cardiovascular regeneration in space. We're in Houston. NASA is here, of course, as you know. So we got some great NASA scientists who are talking about their work to extend our knowledge of the cardiovascular system in space. And we're actually working with them to send a microfluidic vascular avatar up into space to look at the effects of radiation, et cetera. And it's going to be a lot of fun. We got some great speakers. Biomaterials for cardiovascular regeneration is a topic. We're going to have sessions to meet the FDA and meet the dean of the EnMed program. We've got updates on clinical trials. So we're taking our knowledge of biomedical engineering in the heart and actually translating that into the clinic. And we got some of the leaders in clinical trials and cardiovascular regeneration. Josh Hare from University of Miami, Wolf Zimmerman, Joe Wu, and we will have genome editing and cardiovascular regeneration. And there we're going to have Dr. William Poo speaking. And I think I'll introduce him in just a moment, but just to show you the rest of the program, we have a vascular symposium session rather. We have insights gained from cardiovascular fate, changes in cell fate. We have nanomedicine for the treatment of cardiovascular diseases. And then we have, in that final day, we have the Costas family has supported our cardiovascular nanomedicine work, and we're going to be hearing about the latest advances in cardiovascular nanomedicine from the scientists who are in that program. If I could, you can change that screen. I have my crew just take that slide off so we can introduce our guest today, Dr. William Poo. Bill, thanks for joining us today.
Oh, thanks for inviting me. It's an impressive setup you have with your production studio.
Yeah, I've got a great team here today with us. And we've got a great speaker. Dr. William Poo is professor at Boston University in the cardiology department there. And he has done most of his training there. He was a medical student at Harvard and then did his internship and residency and cardiology fellowship training at Boston Children's Hospital and became a faculty member there and now is professor and runs basically the basic and translational research going on in cardiovascular medicine at Boston Children's Hospital. So Bill, I'm looking forward to hearing your insights today, but before we go to your talk, tell us a little bit about your career and what it's been like to be working at the Boston Children's Hospital.
Oh, it's been fantastic being here at Children's and at Harvard. Children's is among the premier research centers is for pediatrics, and they really prioritize research. So it's great to be in that environment. Certainly supported my career, and of course, it's hard to leave this place just because of the number of the intellectual richness and the number of collaborations that you can have.
Yeah, it is a great place, wonderful place. I was there for 3 years myself, and just a mecca of medical research and science. So it's really, really wonderful environment that you're in. And you've leveraged that to do some really exciting things in cardiovascular regeneration and cardiovascular development. I know you're not going to be talking about this today, but I enjoyed the work. I played a minimal role in it, but we made the RNA for your regenerative therapy for myocardial ischemia. Tell us a little bit about that and any new progress in that area.
Yeah, I collaborated with, Lior Zangi was the postdoc that led a project where we used modified RNA to induce the first example of modified RNA-induced organ regeneration in a solid organ. So there we used mod RNA to express VEGF. That was in collaboration with Ken Chen, and showed that improves outcomes in myocardial infarction. And later, the work we did with you, we extended that to use modified RNA to express YAP to promote regeneration in the heart.
Mm-hmm. And what do you think? Where's the field going with respect to modified messenger RNA for cardiovascular regeneration? What are the opportunities? What are the hurdles?
Well, of course, the power of the system is incredible. That you could essentially make a drug out of an RNA and get the biological activity of RNA. I think the biggest challenge is delivery, as they say, delivery. Being able to deliver systemic delivery, particularly of modified RNA, I think, to the heart would be a game changer.
Yeah, absolutely, and there is a lot of work going on in that arena. We have a nanomedicine department here, and there's some activity toward organ-specific delivery of lipid nanoparticles because the RNA, as you implied, the RNA actually requires a carrier, because the RNA itself is so unstable. Messenger RNA is actually designed to fall apart. It's supposed to be a transient message to the cell to produce a protein. You don't want to keep producing that protein. You want to be a transient signal, and so it falls apart. One of the things we're doing to address that issue of the stability of the messenger RNA is one of our faculty has developed a circular RNA for therapeutic purposes. That might have better application for regenerative purposes, where you need sustained production of a protein. But looking forward to seeing more from you in that arena. How many postdocs do you have working with you? Is it primarily, are they physicians, scientists, or are they PhDs? What's the composition of your laboratory?
We mostly have PhD scientists. We have about, say, 12 PhDs, postdocs, and several, like 5 research assistants.
Mm-hmm. So you do a lot of mentoring. What are the most important messages that you give your students and your trainees?
Yeah, I mean, I think to do rigorous work, original work, try to be creative, but also try to make sure the work is, accurate and can be reproduced. I think those are the main messages I try to give.
Yeah, that's good advice. These days it's been difficult for a while. It seems like it might be getting even more difficult with NIH funding. Do you have any thoughts about that?
About how
how to
success with
navigate.
that
Yeah, what do you
funding. I
tell
mean,
your?
if someone has the answer to that, I think that I'd like to know too. But no, I think innovation is rewarded. Also um, NIH has this. They're renewing their emphasis on rigor and reproducibility, and I think having a good reputation in that arena is also good. But I feel like yeah, doing work that's creative and original is probably the most rewarded.
Yeah, absolutely, absolutely. The other thing that I talk to my folks about is trying to diversify the portfolio of funding, because NIH is tough. You can be innovative and have fresh ideas, and sometimes takes some time for people to accept those innovative ideas. And in the meantime, you need to keep things going with other approaches. And so we have American Heart Association.
On the innovation end, you can't be too risky, and on the translational end, also it can be difficult. So other mechanisms to support those efforts are also important.
Yeah, absolutely. So I'm sure you're doing the same thing. We've got NGO support, we've got support from industry and support from philanthropy, so a balance of different funding sources to try to keep the work going. You got to have fuel for the fire. Well, speaking of fire, you got a great presentation for us now, so maybe without further ado, we'll turn to your exciting work. And I've been following your work for quite a long time now in development, the mechanisms of cardiovascular development and cardiovascular generation. So let's hear about that now. If you could bring up your slides.
Okay,
there we
fantastic.
go. Perfect.
Yeah, thank you for this opportunity to present the work. So I just have a few disclosures, including some of the gene therapy approaches that I'll talk about in this presentation. So I'm dividing this talk into two parts. One is about functional genomics and our approaches to understanding the genetic architecture of congenital heart disease. And the second part of the talk, I'll talk about our work on understanding the development of the cardiac dyad and how some of that could be translated to potentially improve heart function. So we're all familiar with cardiac development, going from the simple linear heart tube to complex four-chambered heart. Initial steps involve specification of the cardiac lineages and morphogenesis, but by E14.5, we have a four-chambered heart, which then subsequently grows quite a bit to the adult structure, initially by cell proliferation, and then postnatally, at least in mouse, by hypertrophy. And during this time period, in addition to growth, the cell cardiomyocytes in particular are undergoing quite a bit of development and maturation. And that includes changes in gene transcription, in cell size, nucleation, growth, metabolism, and ultrastructure. And all of these changes are required to make the highly specialized contractile cardiomyocytes that are needed to sustain pumping throughout the life of the organism. So one of our efforts is to understand the epigenetic and translational mechanisms that underlie cardiomyocyte maturation. So one of our efforts was essentially a multi-omics approach to look at changes in RNA expression, histone marks, open chromatin, transcription factor binding, coactivator binding, and chromatin conformation throughout heart development. So our study was published, one study in this arena was published in 2019, where we mapped the location of the major cardiac transcription factors in the heart using this knock-in biotinylation strategy, which we find to be much more reproducible and sensitive than traditional chip sequencing. And from this, we established a map of transcription factor occupancy in fetal, this is E12.5, in adult hearts. And it's striking the differences in transcription factor occupancy between these developmental stages, which perhaps given the, profound differences between these stages of myocytes may not be entirely surprising, but you can see that the majority of binding sites of each of these factors is stage specific. For a long time, the specificity of transcription factors has been in part attributed to their interactions with each other and collaborative binding. So we asked the question of, do we see evidence of collaborative binding in cardiomyocytes? And the answer was yes. So about 50% of transcription factor binding was as a solitary factor, but the other 50% was in collaborative complexes. And we, by looking at ATAC sequencing signal, as well as H3K27 acetylation, which is a mark of active enhancers, we think that the more transcription factors that bind to a region, the more likely it is to be an active cis-regulatory element. Yeah, so not only does the collaborative binding drive cardiac enhancer activity, but also we saw evidence that it supports the specificity of the enhancer. Another thing that we noticed is that many of these multi-transcription factor binding sites were actually H3K27 acetylation negative, suggesting that they had enhancer activity that was not dependent on H3K27 acetylation. But that was prediction of enhancers based on chromatin marks. But there's a gap of going from the chromatin signature to actually measuring transcription enhancer activity. The gold standard for that had been, and I guess the gold standard still is transient transgenesis. However, that is quite low throughput, and at this point, for all enhancers, there have only been a few thousand regions that have been studied by transient transgenesis. So another approach that's much higher throughput is called the massively parallel reporter assay, MPRA, which we implemented as an AAV so that we could deliver it to cardiomyocytes in vivo. So here I showed the structure of an AAV with a reporter construct that has a minimal promoter driving a reporter gene and an enhancer that's in the 3' UTR. So this enhancer not only drives its own expression, the expression of the reporter gene, but also the enhancer is present in the 3' UTR of the reporter. So using this design for a massively parallel reporter assay, we cloned 2,700 candidate enhancers that we identified from the prior work that was either H3K27 acetylation positive and negative for transcription factor binding, meaning the cardiac transcription factors we analyzed, or that had 5 or more transcription factors present with or without H3K27 acetylation. So we delivered this AAV library to neonates and then measured enhancer activity in the 1-month-old heart. And we define enhancer activity as the frequency of the enhancer presence in the RNA compared to the DNA. So this is what the result looks like. We define the active enhancers as the ones that have greater activity than 95% of our negative control regions. So here you can see the negative control regions are in the mostly in the inactive area, whereas many of the candidate regions are active enhancers. And if we quantify the activity of these different groups that we tested, you can see that the H3K27 acetylation regions that do not have co-occupancy by the cardiac transcription factors generally had low enhancer activity. Whereas regions that were bound by 5 or more cardiac transcription factors had high activity, And the level of activity was not dependent on whether or not they had H3K27 acetylation. Again, supporting the idea that some active enhancers are defined by multi-transcription factor occupancy and not by H3K27 acetylation. We took another look at developmental changes in enhancers by using the same biotinylation strategy to look at the genome-wide occupancy of P300, which is a co-activator that decorates enhancers. So here we did a higher resolution view where we measured P300 genome-wide occupancy at several developmental stages. And here we show the dynamic changes in enhancers that occur. So here we defined a set of static enhancers, a set of enhancers that goes down with development, and a set of enhancers that goes up during development. So we again use the AAV-MPRA to validate these dynamic enhancers. So we cloned our enhancer pool into a similar vector and transduced hearts at different developmental stages and measured activity at 3 time points. So here's an example of enhancer activity measured at P7. So again, our positive controls are positive and the negative controls are negative. And then here's our candidate enhancers, some of which were positive in this assay. And if we took the regions that were positive and measured their activity over the 3 developmental time points, you can see that the enhancers that were classified as early P300, meaning that they were active early in development and not in adults, their activity declines over time. Whereas the late P300 enhancers, their enhancer activity increased over time, validating that these are developmentally regulated enhancers. So we looked at the motifs that are driving these developmentally dynamic enhancers, and particularly focused on the ones that are active in adults, and used as background the neonatal enhancers. And the motifs that really stood out were the nuclear receptor motifs. So, for example, the thyroid hormone receptor, which is a well-known driver of cardiac maturation. So, and the role of nuclear receptors in cardiomyocyte maturation has been established by several studies. So we wanted to test this experimentally and to identify the regions that actually require the nuclear receptor motif. So we studied wild type and mutant pairs in which each region was centered on a nuclear receptor that we deleted in the mutant version. And we asked which of these lose activity or change activity with the mutation. So we show the result as a volcano plot. So here's the full change in activity, and the significance level is on the y-axis. You can see that most nuclear receptors that change the activity, when you delete them, they reduce the enhancer activity, suggesting that for the most part, the nuclear receptors are acting as transcriptional activators. The size of the circles here is the strength of binding by the thyroid receptor hormone. So you can see that many of these developmentally regulated enhancers are dependent or bound by the thyroid hormone receptor, as shown by these big circles. But there are also many examples that are, that where the nuclear receptor is important, but is not bound by the thyroid hormone receptor, suggesting that a different nuclear receptor is active at that site. We also looked at chromatin conformation, and just for the sake of time, I'm only going to show one example of finding we had with the chromatin conformation changes over development. This is an example. Here's this is a high C plot. So the red signal shows the strength of interaction of a genomic locus. So for example, the red here is showing the strength of interaction of this region on the genome compared to this region on the genome. So when you sum up all those interactions at E12.5, you have this interaction map. And the blue line highlights a domain of where there's increased interaction within the domain, a so-called TAD. And what the figure is showing is that in the adult heart, you don't have one TAD. Instead, the TADs split, and you get two TADs. So we call that a TAD split. And collaborating with Kai-Fu Chen, who developed software to detect TAD splitting and merging, we found that during development, TADs, we found many examples of TADs that split, but TADs that merge during development are actually quite rare. And in terms of gene expression, this is significant because we can see that the difference in enhancer signal between the sides of a TAD that splits increases during development, suggesting that the splitting is letting one side of the TAD become more active and not the other side. And that correlates with an increase in the difference in gene expression between the two sides of the TAD. So this suggests that developmental changes in chromatin contacts contribute to cardiomyocyte maturation. So in summary, we made a map of transcription factor binding, histone modifications, enhancers, and chromatin contacts during development. We experimentally validated thousands of cardiac enhancers and showed that many are developmentally regulated. We implicated nuclear receptor motifs, including the thyroid hormone receptor. And we showed that chromatin conformation changes are also important in cardiomyocyte maturation. So next we turn to understanding the role of non-coding variation to congenital heart disease. So quite a bit of work over the last decade has elucidated the genetic structure of congenital heart disease. We currently understand about 45% of cases of congenital heart disease and can assign them a dominant role, many of which is genetic. But that leaves 55% where we cannot classify the cause, whether it's genetic or not genetic. And we hypothesize that some fraction of this unknown part is due to non-coding variation.
Bill, can I stop you here for just a moment? Since we're talking about congenital heart disease, trying to make a link to one of the things you just told us. You talked about the importance of nuclear receptors in activating the cardiovascular development program. And one of those nuclear receptors is a thyroid hormone receptor. And I seem to remember from medical school that there is a condition where low thyroid causes congenital abnormalities. I think it has the unfortunate term of cretinism, but is that associated with cardiovascular defects as well, that low thyroid state in newborn?
I believe it's associated with cardiomyopathy, but I do not know if it's associated with a structural malformation such as transposition or hypoplastic left heart syndrome. Mm-hmm. So I think it's more, and that would be consistent with what we found, that it's not during the morphogenesis stage of heart development that we saw that nuclear thyroid hormone being important, but it's in the neonatal maturation stage.
Okay, that makes sense. Yeah, thank you for that.
Okay. The PCGC, among other groups, has generated whole genome and whole exome sequencing data of up now several thousand patients and their parents. And an initial study of the whole genome sequencing data demonstrated an increased genetic burden of non-coding variants in patients with congenital heart disease. So of course, to find so in each individual, there's about 70 de novo variants. Whether or not you have congenital heart disease. So sorting through this level of variation to identify causative variants is, of course, difficult. In total, out of these 750 trios, there were over 50,000 de novo non-coding variants identified. So if you look at all variants, there's no increased burden in congenital heart disease patients. But if you can prioritize the variants based on computational predictions of heart enhancers, then you can see an increased burden of de novo non-coding variants in congenital heart disease. However, this study was very important in showing for the first time that statistically it's likely that non-coding variants contribute to congenital heart disease. However, the study was quite limited in actually testing that with individual variants. So we set out to functionally analyze non-coding variants to identify those that might change enhancer activity. So starting from the same set of CHD trios, there were 57,000 non-coding de novo variants, which we prioritized based on the 3 criteria shown here, to come up with a list of approximately 6,500 candidate variants. We then put those, the reference and the alt versions of those regions into our MPRA vector, this time in the form of a lentivirus. And we delivered that to human iPS-derived cardiomyocytes and measured enhancer activity with the MPRA assay. And the result is shown here. So one axis shows the activity of the reference allele and the other the activity of the alt allele. And colored are the regions that were significantly different between alt and ref. There were approximately 400 variants that showed a significant difference, with the red ones being decreased in the alt allele and the blue ones being increased in the alt allele. And I think it's striking that 50% of the variants increased enhancer activity and 50% decreased it, which might be different from my original conception is that most pathogenic variants would be decreasing enhancer activity, but this might suggest that a significant proportion are actually increasing enhancer activity. If you look at the activity of these regions in more detail, you can see that the ones that increased activity in the reference version, was basically no activity or little activity. And then the alt allele created an enhancer out of something that was not initially an enhancer. Whereas the decreased activity, they had activity in the ref allele that was lost in the decrease in the alt allele. So we wanted to validate this further, and we felt the gold standard test would be to make knock-ins of these alleles in iPS cells, differentiate them to cardiomyocytes, and then measure gene expression of the cardiomyocytes. So we made 10 knock-in lines of candidate variants that had effect in our MPRA. And this was an enormous amount of work. For each variant, we isolated a minimum of 2, usually 3 different lines. And for each line, we differentiated them at least 3 times. And from that, we found 4 variants that consistently affected the expression of nearby genes. So I'll just quickly show the results here. So this is a really interesting result. BCOR is an epigenetic repressor. The variant disrupts a SMAD motif and decreases the expression of BCOR. Myocardin is a well-known transcriptional activator. So this variant disrupted a motif nearby myocardin and also created a TBX motif with the variant decreasing myocardin expression. So we wanted to test these 4 candidates further to see how much they changed gene expression. And to do that, we used multiplexed single nucleus RNA sequencing. So here we took this set of variants, we differentiated them into cardiomyocytes, then we did single nucleus RNA sequencing, and here we're plotting the result. This is just showing all iPS cardiomyocytes. We identified 8 different clusters of them. If you look at where the wild type ones are, they're mostly in cluster 1. And strikingly, the BCOR iPS cardiomyocytes are primarily in clusters 2 and 3. And the myocardium variants are highly enriched in cluster 2 as well. So you can look at that with this stacked bar plot and see that these variants do significantly affect the distribution of cardiomyocyte states that differentiate from these cells. In contrast, if we look at some negative control regions that did not affect the MPRA assay, they did not significantly change cell state. So to summarize, we used the lenti-MPRA to test about 6,500 CHD-associated non-coding variants and found 400 that were classified as damaging. And about half of those increased enhancer activity. We further validated 4 out of 10 and showed that they significantly change iPS cell cardiomyocyte transcriptional state. So together, these data suggest that these non-coding variants participate in the pathogenesis of sporadic CHD. Oops. So I'm going to turn now to the second part of the talk. I guess we don't have that much time left to talk about cardiomyocyte ultrastructure and how our studies of that led us to find a potential treatment for heart disease. So a hallmark of mature cardiomyocytes is the dyad. The dyad is where the junctional SR becomes closely opposed to T-tubules. On electron microscopy, T-tubules are seen as this donut-shaped ring that's adjacent to Z lines. And you can see that if you zoom in, that the junctional SR is like a pancake that's on the side of the T-tubule. And they have, within 10 nanometers, they have this cytoplasmic cleft that's contiguous with the cytosol. So this is an amazing ultrastructure, incredible organization in which the T-tubules penetrate the cardiomyocytes and become closely opposed to the sarcoplasmic reticulum. This structure is essential for efficient calcium-induced calcium release and coordination of cardiomyocyte contraction. So to understand that a little more, I'll just go through the cardiac calcium cycle. So an action potential will penetrate the cardiomyocyte down the T-tubule and then activate the L-type calcium channel. That lets a little bit of calcium come from the extracellular space into the dyadic cleft, which then activates the ryanodine receptor that's sitting adjacent on the sarcoplasmic reticulum. This releases calcium from the SR into the cytosol, and that induces contraction of the sarcomere. And I think this is where the alignment of these dyadic structures along the Z line positions the calcium release unit so that the release of calcium is consistent at different sarcomeres, enabling coordinated contraction. So what orchestrates this precise nanoscopic assembly of dyads? Quite a bit of work by others has shown that these factors, JPH2, BIN1, and we also showed the ryanodine receptor, are important in regulating T-tubule organization. However, what positions the T-tubule, what positions the ryanodine receptor at the Z lines was not known. And in fact, the ryanodine receptor positioning predates the formation of T-tubules. So for example, atrial cardiomyocytes don't have many T-tubules, yet they have highly organized ryanodine receptors positioned in the same way at the Z line. The same with the P7 ventricular cardiomyocyte, which is prior to the stage at which T-tubules develop. They also have highly organized ryanodine receptors. So this shows that you don't even need the T-tubule to position the ryanodine receptor properly. We looked at E15.5, and this is when, so sarcomere assembly, so the white staining is the, here is the Z line. And you can see here that this set of Z lines does not have organized ryanodine receptor. Whereas if you look at this set of Z lines in the same cardiomyocyte, they are starting to assemble the ryanodine receptor at the Z line. So this shows that the first thing to form are the Z lines that forms the organizing scaffold that then positions the ryanodine receptor. So we asked, what is it that is positioning, what is mediating this positioning of the ryanodine receptor? And to find this factor, we used proximity proteomics to screen proteins that are associated with the ryanodine receptor. And so this just summarizes the result of our proteomic screen. There are many known members of the dyad in this screen, but one factor that has been very little studied that we found with this very interesting protein CMYA5. And we decided to look into this protein further. So first we used the proximity ligation assay to confirm that CMYA5 is located next to the ryanodine receptor. So the PLA signal is positive when the two proteins are within 40 nanometers. So this shows that these two proteins are very close to one another. And some of the prior work on this protein showed that in other cell types that the ryanodine receptor does interact with the CMYA5. So we made a CMYA5 knockout mouse, and we showed that this knockout disrupts the organization of T-tubules as well as the positioning of the ryanodine receptor. And that's also seen here on electron microscopy, where the T-tubule is, the morphology is disrupted and its location with respect to the Z line is also disrupted. So we also looked at these cardiomyocytes that don't have T-tubules yet in the knockout. And again, atrial myocytes lost their ryanodine receptor organization, as did the early ventricular myocytes that do not yet have T-tubules. And in the embryonic cardiomyocytes, we found these cardiomyocytes that had CMYA5 that are already at the Z lines. And so some of these do have organized ryanodine receptor. That's the yellow arrows. And then the red arrows show the CMYA5 at the Z line lacking the ryanodine receptor. We almost never saw a ryanodine receptor at the Z line without CMYA5. So putting that together, we came up with this model in which CMYA5 acts like a tether that binds to both the Z line and the junctional SR ryanodine receptor. And then in collaboration with other proteins, later the T-tubule is formed. So we also looked at calcium handling, which is dependent on the dyad structure. We found that the mutant had increased abnormal calcium release events and also decreased amplitude of the calcium transient. You can also see that the coordination of the release of calcium is very precisely timed to pacing spikes in the wild type, but becomes more ragged in the knockout, perhaps accounting for the reduced size of the calcium transient. So the knockout also has a mild cardiomyopathy. So by 3 months, you can see reduced fractional shortening. And then this becomes quite profound in a TAC model where, so we apply pressure overload, and then the knockout mice have quite severe cardiac dysfunction. So to summarize this part, we showed that CMYA5 tethers the ryanine receptor and the junctional SR to the Z lines, creating the initial positioning of the dyad. And that loss of CMYA5 impairs this organization, and it reduces coordinated calcium release and heart function, particularly in the setting of pathological stress. So finally, in the last few minutes, I want to show how we took advantage of this to find a potential therapy that may improve heart function. So we know that in human heart failure, the dyads become disorganized. That's been particularly well studied with T-tubules. However, it's also true of the junctional SR that becomes less organized. Here's an example of myocardium that we stained for the ryanine receptor, which shows some lack of organization in the dilated cardiomyopathy. And there's some other studies showing the same. For example, here they showed that there's actually less ryanine receptor organized around each myofibril. We did find for CMYA5 in heart failure, we did not see a change in its localization. It still stayed localized to the Z line. The mRNA levels were not changed. But if we look at a Western blot of CMYA5, it's a little bit messy. There are multiple bands, and these bands go away in the knockout, so we think they're all related somehow to CMYA5. But only this isoform here co-IPs with the ryanine receptor. And so we quantified this band and found that it's decreased in TAC. We saw the same thing in human heart failure. And so we asked if there was a decrease in CMYA activity in heart failure and if that could contribute to cardiac dysfunction. So to answer that question was a little complicated because CMYA5 is actually a huge protein. It's 3,700 amino acids long. So it's too long to be expressed by AAV or even adenovirus. So we looked at the structure of CMYA5 and we noticed that the middle portion of CMYA5 is unstructured and it has very poor conservation, whereas the N-terminus and the C-terminus are better conserved. So we divided the protein into these 3 parts and we studied either each part separately or the fusion that omits the middle part. So we showed that either protein that contains this C-terminal part, which we labeled MD9, it interacts with the ryanine receptor, whereas the N-terminus does not. On the other hand, the N-terminus is sufficient to localize to the Z lines, whereas MD9 does not. And if we look at rescue of CMYA5 activity as defined by positioning of the ryanine receptor, you can see that neither MD9 nor the N-terminus is able to rescue. But the fusion of the two, in which we call mini CMYA5, is sufficient to rescue the RYR2 positioning. And in addition, the fusion is sufficient to rescue T-tubule formation. So we asked if this mini CMYA5 could rescue heart function in mice. So here, this is in a TAC experiment. So we took the CMYA5 knockout mice and either did sham or TAC. At 0 weeks, we saw that the mini CMYA5 improved the function of the knockout animals, and then at 8 weeks had very poor function again in the control treatment and somewhat improved function in the TAC animals. So at least in the CMYA5 knockout background, mini CMYA5 does have a rescue effect. And that also seen here in the structure of the T tubules and the ryanine receptor localization. But the big question, what about wild type mice? Can we improve the function of wild type mice using mini CMYA5? So again, we did sham or TAC, and so sham TAC animals treated with the GFP control had dysfunction at 8 weeks, which was substantially improved by the mini CMYA5. And you could also see that at the level of T-tubule and junctional SR organization, that the mini CMYA5 improved the organization compared to the control. So in summary, one last point. We also looked at iPS cardiomyocytes, which, as everyone knows, lacks dyads. They lack T-tubules, and the ryanine receptor is not localized to the Z lines. One big reason for this may be that iPS-derived cardiomyocytes actually have very little expression of CMYA5. So we patterned cardiomyocytes so that they had well-organized sarcomeres. And then we looked at ryanine receptor organization with LAC-Z or with mini CMYA5. And I hope what you can see here is that in some of the some areas of these patterned cardiomyocytes, we actually did get RYR2 organizing at the Z lines. So unfortunately, we were not able to detect formation of T-tubules in these cardiomyocytes. So it seems that CMYA5 can promote RYR2 organization in iPS cardiomyocytes, but it's not sufficient to induce full dyad formation. Okay, to summarize, we developed a mini CMYA5 that both localizes to the Z lines and organizes RYR2. This is able to complement the CMYA5 knockout, and it stables the junctional SR and T-tubules during pressure overload. So there may be applications for this for treatment of systolic cardiac dysfunction. Okay, these are all the people that did this work. Particularly thankful for Fujian. Actually did all of the work on CMYA5, and he recently left the lab to start his own lab at Fudan University. And this group here did the enhancer work in the myocytes, and Feng led the work on the congenital heart disease variants. Thank you.
Thank you, Bill, for a really exciting talk. And also thanks for being such a great mentor. You mentioned Fujian Lu is the fellow who did most of the work and is now going to be a faculty member at Fudan University. I think that is a mark of a successful laboratory when you have your proteges going to great universities and carrying on the work, extending the work that they did with you in the lab. We're running to the top of the hour, but I do have a couple of comments and questions for you. One comment is that, boy, it'd be great to work with you again. The mini CMYA5 didn't appear that it fully reversed the abnormality in the human iPS cardiomyocytes. And I wonder if the full length, you'd have better effect with that. Now I know you couldn't put it into an AAV, but we actually have experience now in making larger mRNAs. So we make self-amplifying RNA, and those are, 9 to 11 kb. And if my calculations are right, your CMYA5 is probably in the range of 8 to 9 kb. So I think we could certainly take a crack at that for you. We've got better T7 polymerases now, and we're getting much less abortive transcripts, so we could help you with that.
Yeah, that'd be exciting. They certainly transduce IPS cardiomyocytes very well.
The other thing is that Francisco Altamirano here has a very nice engineered heart tissue system where you could potentially extend some of your work to human heart tissue and look at effects on electrophysiology and contractility, because of course, as you well know, the mouse heart is very different from the human heart.
Yeah, that'd be great to do. We also are using some engineered heart tissues based on the Zimmerman platform, and Eschenhagen and Zimmerman platform.
Great. We have some questions from the audience. So one question is, what's your opinion on engineered heart tissue? I think it's kind of something I just alluded to study the cardiomyocyte in a more mature condition.
Yeah, they're clearly more mature than IPS cells plated in a monolayer on a rigid substrate. They still don't have dyads, so they're not, I wouldn't call them a fully mature myocyte. But they're, depending on what the question is, they are an excellent platform. Yeah. We use them all the time.
As a vascular biologist, I have to ask this question. Would your cardiomyocytes be benefited from having an endothelial association? What's the interaction there?
Yeah, many groups have looked at that. Whether adding endothelial cells to engineered heart tissue or cardiomyocyte cultures improves function, and I think the general result is yes, it does have a good effect.
Does it have an effect, Bill, on the electrophysiology? Do you get more mature electrophysiological patterns from your IPS cardiomyocytes?
We have not looked at that ourselves, but I believe other groups have. If I remember right, Christine Mummery had several papers on that and showed that there is an effect, a beneficial effect on the electrophysiological properties as well.
Yeah, great. Well, I really love to see the basic science starting to get translated toward clinical therapeutic, and we'll follow your work with CMY5 with interest to see how that develops, and if there's something we can do to help you, just give me a call. Well, thanks, Dr. Pu, for joining us today and showing us your exciting science, and I want to thank the audience, the attendees, for joining us at the cutting edge of cardiovascular sciences. Thanks again, Bill.
Thank you. It's my pleasure.